Waving a pattern of lines in front of the cat's eye to help me find visually responsive neurons. Compared with retinal ganglion cells, cortical cells have rather little spontaneous activity. What you heard was some action potentials growing out of the background noise as the electrode approached a cortical cell. This neuron clearly responds to something on the patterned card. Unlike retinal ganglion cells and the cells of the lateral geniculate nucleus, cortical neurons almost never respond to changes in general illumination. Off, on. You can hear that there's no response. So now, let's use patterns of light on a television screen in front of the cat to try to define the cell's receptive field. This is a so-called simple cell. It's thought that these neurons receive direct signals from a fairly small number of incoming fibers. Even simple cells don't respond vigorously for small flashing spots, but they will always fire for a long bar of light. Now let's look at the response for a flashing bar in different parts of the receptive field. On, off. In the middle of its receptive field, this cell gives on responses. It produces a burst every time the light is switched on. We indicate that on response with plus signs. Now let's try the areas flanking the on zone. Here it produces a burst of impulses when the light goes off. We plot the off zone with minus signs. And on the other flank, there is another area of off responses. So this receptive field is rather like that of a ganglion cell or a cell in the lateral geniculate nucleus, except that the on and off areas are not round, but are elongated. Now I'm going to try changing the orientation of the bar. On, off. There's no response at all because the light falls simultaneously on excitatory and inhibitory zones. On, off. So this sort of cell will only respond if it's shown an edge or bar of the correct orientation. These cortical cells are called orientation detectors. The response is usually even better if the bar is moving. First, at the correct orientation. Now, at slightly different angles. And now perpendicular to the best orientation. So this cell responds best to an almost vertical object. Other neurons prefer other orientations, some horizontal, some diagonal, and so on. Let's search for another cell. This one is a complex cell. These neurons are thought to be much more complicated in their connections. Their receptive fields are usually larger than those of simple cells, and they are often spontaneously active. Again, this cell refuses to respond to overall changes in illumination, and like many complex cells, it does not even respond reliably to a flashing bar of light. However, complex cells always respond well to a moving target of the correct orientation. You can see that this one responds for a roughly horizontal bar, but only if it's moving upwards, not downwards. So this particular cell, like many others, is direction selective, as well as being sensitive to the orientation of the target. There is a third class of cortical cell we can find, and because their properties are so complicated, they're called hypercomplex. They again respond to a moving bar, but not only must it be of the correct orientation, it must also be of exactly the right width and length. This hypercomplex cell responds to a diagonal bar moving downwards. But look what happens if I increase the length of the bar. The response is inhibited if the pattern is too long. And now let's try again with the optimum stimulus. So the receptive field must have strong inhibitory areas at each end that make it respond only to an edge or bar of a particular length. Sometimes there's an inhibitory zone only at one end of the receptive field, so the cell responds best to a 90-degree corner. This specificity for the length of the target is the defining characteristic of hypercomplex cells. So cortical cells are orientation detectors. Simple cells respond to flashed bars or edges and have straightforward receptive fields. Complex cells respond to moving bars, and hypercomplex cells to bars of a particular length. Experiments with microelectrodes also tell us something about the microscopic structure of the brain. They show that the visual cortex is organized into blocks or columns of interconnected cells. The incoming fibers enter the cortex at the base of each column, and here, in layer 4, are many simple cells. Complex and hypercomplex cells are usually found in the surrounding layers. If a microelectrode is driven down perpendicular to the surface through a single column, every neuron is found to respond to the same orientation. If the microelectrode penetrates diagonally across many columns, there are sudden changes in preferred orientation from the cells in one column to those in the next one. The columns are about 0.5 millimeter across at the surface. David Hubel and Torsten Wiesel at Harvard, who first described the properties of these orientation detectors, have suggested that within each column, the simple cells send fibers to complex cells, which in turn transmit excitatory and inhibitory messages to hypercomplex cells. In this way, all the neurons in a single column respond to the same orientation. Remember that only one eye was uncovered throughout this experiment, although in fact most cortical cells receive similar signals from both eyes.